Hello there. My name is Ajay David, and I'm a PhD candidate in the lab of Dr. Michel Caillouet at McGill University, Montreal, Canada. And I'm filming myself to answer the question, what excites me most about my research? To answer this, I thought I'd provide an anecdotal story that happened a couple of weeks ago. I was swimming at a lake here in Montreal, and I nearly tripped and fell and hit my eye, or close to my eye rather. And this was a rather traumatic and scary incident for me because if I were to have hit my eye, I could have potentially lost my vision. And indeed, millions of us live with certain forms of vision debilitating diseases and the more permanent form, which we call it blindness. However, ultimately, the core pathology behind blindness is the loss of retinal neurons and the inability of our own body to recover these retinal neurons following this loss. However, this feature that we call blindness is not something that's shared by all seeing organisms. For instance, certain species that lived in that lake, certain species of fish and frogs, have the remarkable capacity of regenerating their eye throughout their lifetime. And this is driven by the glial cell in the eye known as Mueller glia, which in fish and frogs have the stem cell ability to become retinal neurons, but in mammals, both humans as well as the model organisms that I work with, mice, have lost this during the evolutionary process. And so the overarching goal of my PhD research is to uncover why this is the case and drive regeneration in mammals. I believe that we can sort of uncover this by looking both at the differences between fish and mammals, but also by looking at how development occurs and studying these conserved development principles and see if they can sort of regenerate the eye. And why this is particularly interesting to me to attend the MIT's Koch Institute's Cancer Biology Research Day is because I believe that regeneration and cancer are two sides of the same coin. They're both intrinsically, intimately related to each other, and and ultimately, I want to sort of pass out how we can uncover these conceptual mechanisms in both cancer and regeneration. For instance, in cancer, we are not interested in cells becoming proliferative and we're trying to stop them from de-differentiating and so on. But in regeneration, it's the complete opposite. We want the cells to proliferate. We want these dormant cells to de-differentiate. And so it's almost the opposite that we're trying to achieve. Indeed, for instance, the eye, retinoblastomas are a relatively rare form of cancer. And so the ability for the eye to sort of like regenerate is a evolutionarily hard mechanism that has implications, especially when we try to uncover the regenerative principles. And so in my belief that these concepts are intimately related, the regenerative principles as well as cancer biology, I believe that attending the MIT's Research Day could be both beneficial to myself and to my project, as well as understanding these deeper concepts in the forefront of cancer biology, which may ultimately be very beneficial to unlocking how regeneration occurs in tissues that have no regenerative capability and have evolved to sort of stop this. And ultimately, if we can understand how regeneration stops, we can also use that in cancer to sort of stop cancer from forming and initiating at the start. So with that, I'd like to end and thank you for listening to my 3-minute video. Thank you so much.